For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 3, Episode 43 of Surfing the NASH Tsunami. Our look at the evolving insights about the role of combination medicines in NASH and NAFLD therapy starts now.
This week on Surfing the NASH Tsunami.
When we think about NASH drugs, we're not just thinking about hepatic endpoints, we're also thinking about the effects on metabolic syndrome and cardiovascular outcomes. And semaglutide hits every point. The only missing piece was really efficacy on fibrosis regression. And this has been the Achilles heel. This is the rationale why we needed to look into combination therapy with semaglutide, and we needed medications that have anti-fibrotic effects.
For the first time I saw a map of the liver in the context of NASH, I was struck by exactly how many different places that you can attack. We talk about the idea of taking shots on goal. The problem is the liver has 8, 9, 12, 15 different goals. You're not going to want to shoot 4 pucks at the same place. You're probably going to want to invest in 3, 4, 5 different therapeutic targets.
This is a field of combo, the combo of NITs, staging the disease and monitoring it, and eventually a combination of drug like metformin and SGLT2 in diabetes and metformin and GLP-1 in diabetes.
Even if we get the drug approved, might not have the answer what's the best patient for that drug yet, and I think it'll be important to stay on top of it and be open-minded in terms of how we move these field forward.
The ability to show real-time results to patients short-term really is a motivator to continue lifestyle modification in addition to the medications, and that sounded to me as a huge benefit that you can take into the real-world patient population and get immediate turnaround on their behavior change.
The exciting thing for me is to see rates in the 90% and that 30% relative reduction. I'm hopeful that we get to a point with these combination therapies that we start talking about a cure for NASH in some patients where there's really no evidence of the disease whatsoever, where there's not enough liver fat to call it NAFLD.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researchers and key opinion leaders Professor Jarn Schottenberg, liver wellness advocate Louise Campbell, and pricing and forecasting guru Roger Green, along with this week's guests, hepatology researchers, and key opinion leaders Drs. Naim Alkhouri and Mazen Noureddin, as they discuss new insights on the development and evolving role of combination therapies on Surfing the NASH Tsunami.
First of all, it's great to be back with you guys. I actually was able to get a week where I did not focus on the podcast at all. Thanks to Louise for a fantastic job of hosting, to Jorn for a great job of guesting and dealing with tough and challenging questions, and to particularly Jake and Mike, who put the whole thing together in the back room with a little bit of support from Louise so we could get episodes out, and I didn't have to worry about it. That was a real gift. First time I've been able to do that in 2.5 years. So thank you to all of you, and glad to be back with all of you audience and everybody today. We have a fantastic episode, I think, with 2 of our favorite guests. Well, first of all, let me just say hi to our regulars and then we'll go on to those guys. Louise, how are you today?
I'm very well, thank you very much, and very nice to have you back. So I can chill and relax now, and I'm glad you had a lovely time in an area that I thought was a TV series. There's a program or a film called Jersey Shore.
Oh, there's a lot of stuff called Jersey Shore.
So when you said Jersey Shore, I thought you were just talking about a TV program.
Cape May is actually the least TV-ish part of the Jersey Shore. It should be noted that Louise is all in red today. I don't think that's because Liverpool broke their terrible season start with 9 goals against Bournemouth yesterday, but it might be. Just never know.
No, it's because I've just played golf.
Oh, better. Jorn, how are you today?
Good. I'm out of my vacation for 2 weeks, back into clinics, caught up with stuff, I think, and very excited to be back here with good friends Naim and Mazen and talking about their great publications. They've brought forward some great stuff more recently, and the combination paper is clearly very exciting to me, so looking forward to see and hear about it.
Yeah, I'm on the other end of your thing, which is I now have, 18 days of work to do between here and the end of Friday, but I'm hopeful. Mazen, how are you today?
Good. So I just want to make comment on Jorn's comment, which is he's caught up. I wish I can get that feeling one day. He came back and he's caught up already. I'm here. I'm not caught up since this morning. I have 100 and something email to go through, but it's good to see friends and good faces.
Yeah, I'm going to send you a note later asking if we can find 10 minutes, a text message. It won't be if we can find 10 or 15 minutes to catch up on a couple of things today, tomorrow. But I understand your schedule. We'll do the best we can. And then finally, Naim, who I've come to consider maybe the busiest guy I know the last couple of months. How are you doing?
I'm good, busy as usual, but good to be back. Good to be with you guys. I've had a busy weekend. I was in California and then I came back last night. I went to a Chris Rock concert and I have a few funny jokes, but I don't think we can share on this podcast, but maybe we can catch up later.
Oh, go on. It would open it to more listeners.
I think you should share it on this podcast.
Not the appropriate audience, I would say.
That's exactly why Mazen wants you to share it. He wants to see exactly whether you can blush enough that we will see the blush without having video on the screen.
Yep.
Troublemaker.
We're not that boring in hepatology.
We're not boring at all.
Never were.
No, I never will be. So with that, why don't we just do icebreaker? One good thing in the past week, professional or personal, take your pick. Bold one, go first.
Oh, I'll be bold and jump in. Apart from allowing you to have some holiday, I enjoyed a brief trip to Copenhagen to give a lecture. So it was a nice trip. It was a lovely city. And the hotel was slightly too techno, and it takes you 4 minutes to book your breakfast slot rather than just ask somebody to book a breakfast slot. So sometimes technology doesn't take you all the way there. It can be disruptive, but it was a lovely trip and I enjoyed giving the lecture. So that was my professional and personal, I suppose, for the week.
Fantastic. Next.
I guess I'll go next. So we got some good news last week about our collaborator at the University of Arizona in Tucson with some funding coming his way, Dr. David Garcia, to do more fibroscans in the Mexican-American community in Tucson. And what's unique about this project, and I think, Louise, you'll appreciate this, they take the machine to their home and they scan the whole family and try to identify high-risk individuals. So this is exciting, and we're trying to support also at Arizona Liver Health. Also, there's some funding coming to another collaborator who's working on MRI technology. So good things are happening in terms of funding for NAFLD.
That sounds great. Really, really exciting. And for those who can't see, because we're mostly on audio, Louise did a double-handed fist pump when Naim announced going to the home. Jorn, why don't you go next so that Mazen can swallow what he's chewing on?
Sure. So beyond the usual clinic stuff, serving my patients, we had a patient advocacy group stop by that wasn't related to liver disease specifically, but it was something that we support as clinicians, and they went on a tour bus and actually stopped by our center to support our local patient advocacy groups, which I think is great. And I was happy to, stop by and exchange with them. So that was professionally, I think, something where it was satisfying to be in touch with the patients, even advocacy group. And my highlight of the week was, of course, having a dinner date with my wife on Friday evening. Kids were taken care of, and we spent a lovely evening going out for dinner. That doesn't happen too often.
I understand how that works. I used to be in that boat a while back. Mazen, you're up.
Yeah, I'll go along the same line professionally. As you know, I have now a research institute in Houston called the Houston Research Institute that we opened door in July. This is a private institute in addition to my academic job. But the exciting news that we did our first SIV last week for a study, and we're going to have another 2 SIVs this month. And I got my drawings back from the architect on the extension of this facility from 4,000 to 10,000 square, which will be open in February, the entire 10,000 square foot. So exciting time of NASH and NASH research, and we're on.
And you continue to look good and svelte and like you're taking care of yourself. So I'm hoping that's not a mirage. You look great.
I really like it. So since we're hitting on each other on this podcast, I'll tell you my secret. So one of the favorite actors, not a secret, but something I haven't said before. One of my favorite actors is Matt Damon. I think Schattenberg is the Matt Damon of NASH.
Well, what can I say?
He looks like him.
All right.
Jorn, this is not the first time I hear this, I must say that. So I've heard it multiple times before.
He's in a better shape, and I think a couple years back it was a little closer, but I got to start working on my, got to do more workouts to meet him there.
I kindly ask you, Roger, to keep this online so we spread the word that he's the Matt Damon of NASH.
It's interesting. I will be happy to do that, Mazen. When you said that, I was thinking the other analog is Dorian Gray because he keeps looking younger the more time I spend with him. Between the youth and the workout, podcast. It's really pretty compelling as I watch my hairline recede into the Pacific Ocean from all the way from New Jersey.
No, the highlight should be we should congratulate Dr. Noureddin for opening up this tremendous research institute and getting things started there. This is not about.
Actually, that was where I was going to go next. So thank you. My highlight is just I was away for 2 weeks, which is, I had family with us for a time, friends for a time, and it was just a great time to be away. Coming back, we have a lot to look forward to, and I'll talk about that in the business section. But right now, why don't we just dive on into our podcast? Because as you might have heard, everybody's really busy. So we would like to get started so we can get our information out to you folks in a good and timely fashion and get everybody back to what else they're doing. The subject for this podcast popped up, I think, in May originally when the online version of the paper that Hepatology actually printed, I think this is printing this week, September 1st episode, if I'm reading that right, in JHEP?
JHEP.
In JHEP, that's right, which was a paper taking a look at the combination therapy of semaglutide, cilofexor, firsocostat. Naim has mentioned that on this podcast, I think, once or twice since, or in some setting with me. It might be the podcast because it's the only time we've recorded since then. And it's an exciting and interesting paper, and I think it speaks to an issue we talked about a couple of times, which is different ways to think about combination therapies, not being what they were in the first place, which is if two drugs fail in mono, can I put them together in combo? And turning into, okay, what are all the different things that we want to do? So I'm hoping with the aid of first author Dr. Alkhouri and last author Dr. Noureddin, if I got the orders right on that, and everybody, that we can go through the thinking behind how do you think about which drugs belong together in combination? What can we learn about that from how this study got constructed, and then the results of this study and what you think it portends for the future? So Naim or Mazen, take your pick. Let's start on how did all this start, and where did you start on it?
I'll let Naim take the leading role as he's the PI of the study, and I want to remind everyone of his excellent presentation. I think it was during AASLD, Naim, right? Virtual one. And
yeah.
I was very impressed the way you handled the questions, actually.
Oh, thank you, my dear friend. So Roger, I presented this at AASLD 2020. It took us a while to get this published, but I'll give you some of the background. Obviously, as you mentioned, it's a combination, 3 medications, semaglutide, which everyone knows, GLP-1 agonist, cilofexor, which is an FXR-agonist, kind of similar to obeticholic acid, and then firsocostat, which is what we call de novo lipogenesis inhibitor. So we had the phase 2b with the semaglutide alone that showed great efficacy in terms of NASH resolution and 59%. When we think about NASH drugs, we're not just thinking about hepatic endpoints, we're also thinking about the effects on metabolic syndrome and cardiovascular outcomes. And semaglutide hits every point. I mean, patients lose weight, they improve dyslipidemia, they improve their A1C, insulin sensitivity, and they have, proven cardiovascular outcomes, especially in patients with type 2 diabetes. So semaglutide is a great medicine, but the only missing piece was really efficacy on fibrosis regression. And this has been the Achilles heel. And the fact that, the mechanism of improvement is related to weight loss and not everyone loses weight on GLP-1 agonists. This is the rationale why we needed to look into combination therapy with semaglutide, and we needed medications that have anti-fibrotic effects. So Gilead had a program that had 3 drugs in it, cilofexor, which was not effective, but the other 2 drugs were cilofexor and firsocostat. And we had the Atlas trial that included about 400 patients, all biopsy-proven disease with F3-E43. And in that trial, we noticed that if you look at the combination of firsocostat plus cilofexor, there was regression of fibrosis by 1 stage in about 21% of patients, and in the placebo arm, it was 11%. Now, this did not meet statistical significance just because the study was underpowered and we had arms with cilofexor that had no efficacy. So it was really hard to make conclusive arguments that this combination by itself is going to work. But that was a really high-risk patient population because 56% had cirrhosis. And to see fibrosis regression in 21%, in my mind, this is very clinically meaningful. So this was the rationale why to use the cilofexor, firsocostat plus semaglutide. And then we designed the study, and I'm happy to go through the protocol quickly and maybe give like an overview of the top line results. But this was, everyone received semaglutide, so there was no pure placebo because we wanted to show that the combination had added benefit on top of semaglutide alone. So the first arm was semaglutide, and then we had semaglutide plus firsocostat alone, semaglutide plus cilofexor low dose 30 milligrams, semaglutide with high dose cilofexor 100 milligrams, and then we did the triple combination therapy, so semaglutide, firsocostat, and cilofexor. And we included patients with biopsy-proven NASH, F2, F3, or a non-invasive way to diagnose NASH, which was basically MRI-PDFF more than 10% plus liver stiffness more than 7 kPa. And we did not want to include cirrhotics in the study, so we used the fibro test less than 0.75 to exclude cirrhotic patients. And patients were treated for 6 months, 24 weeks, so a relatively short term. And not to get into too many details, but in terms of weight loss, the weight loss was similar across the groups. Everyone lost about 7 to 8%. But when we looked at MRI-PDFF improvement, we noticed the highest reduction was with the triple combination therapy, so semafir, cilofexor. And if you look at that 30% relative reduction from baseline, which has been used to predict histologic improvement, 92% achieved that with the triple combination therapy versus 80% with semaglutide. And then we even raised the bar, so we said, okay, how many achieved more than 50% reduction from baseline in terms of liver fat on PDFF? And that was achieved in 85% with the triple combination compared to 40% with semaglutide. And then we took it even to 70% relative reduction, and that was about 1/3 of patients with triple combination versus only 7% with semaglutide. So we see clear evidence that the triple combination has added benefit. And then we also saw a reduction in ALT, AST. There was some reduction also in CK18, cytokeratin 18, biomarker of hepatocyte apoptosis. And then we also looked at liver stiffness on FibroScan, and we used a 25% reduction from baseline, and we showed that about 50% achieved this with the triple combination compared to about 35% with semaglutide monotherapy. And then we also looked at the FAST score that includes CAP for liver steatosis, liver stiffness for fibrosis, and AST for inflammation. And we showed reduction in the FAST score at 0.39. Remember, the FAST goes from 0 to 1. So, this is about 30% reduction compared to only 0.19 with semaglutide monotherapy. So again, indicating that potentially the triple combination has higher efficacy. The other thing we looked at was also how did semaglutide help with some of the liabilities that cilofexor and firsocostat have. Firsocostat can increase your triglycerides, and cilofexor can increase your LDL. And we noticed that we mitigated these adverse events by adding semaglutide. And this is one other angle we think about combination therapy. In addition to increasing efficacy, can we mitigate some of the concerning adverse events? So we feel like this combination is hitting on both, increasing efficacy, but also mitigating some of the dyslipidemia issues that we have with firsocostat and cilofexor. So sorry for taking too long, but I've been very involved in this study. This is like my baby, so just wanted to give you the historic background and the overall top-line results.
So first of all, Naim, that wasn't long at all, and that was fantastic. Let me offer other people questions before I ask any. Go ahead. Questions?
Yeah, congratulations, Naim and Mazen. I think an outstanding study, really down the line of combining multiple MOAs to increase the efficacy, as you nicely detailed, on a lot of NITs. But something that was very interesting to me is that you mentioned toward the end was that the safety concerns you might get from the addition of multiple drugs, or it could even be a little bit dampened when you compare it to the individual drugs. So I think this is very appealing when we consider that each drug has a rationale to be used in NAFLD, and each drug has side effects. And it was reassuring that the lower drug, the lower concentration of the FXR, which gives us side effects in terms of, pruritus potentially, or LDL, could be used, and that these changes were even decreased. So I think that's a very reassuring, and to me, that's the powerful message out of this. I think it's difficult to really deep dive into the efficacy differences based on the small groups and the multiple comparisons, but really the strong message is you can combine and you can mitigate the safety that comes up. That was my take, and congratulations again. I think that's really a strong way forward.
Louise, you have anything, or Mazen, you have anything you want to add about the paper?
No, I'd just echo Jean. Congratulations, guys, on that paper. It seemed a wonderful read, and some great information coming out. As Jean said, it's encouraging to know that the side effect profile, although they were there, they seem to be dampened down. That's what patients want. I think, Naim, you mentioned that it was a relatively short study. Is there any chance that these drugs may wane over time, or how long the proposed treatments would be? Because because they did seem very efficacious, certainly in the combinations.
I can take a shot at this. I want to thank Louise and the Matt Damon of NASH for their comments. Actually, the first comment I want to make in terms of the duration of study, I think the study still could have shown also more positive results if it extended beyond the duration. Because as you know, SEMA is a titration up, and it takes about 16 weekish to titrate. So you can argue that you barely reach the therapeutic dose that it's going to be used for. So this is one. On the other hand, as Naim mentioned, there was a significant effect on reduction of the fat. So at one point, it's going to continue to peak, and then you are not going to have much more fat to melt, if you want to say, and the effects will be on inflammation and fibrosis, which we thought that these drugs or the other mechanisms added, not saying that SEMA doesn't do that. I think it will if you use it for a good duration of time. So this is one. I also have a comment on the trial itself and its importance. Well, you can say this is open label, there's no placebo arm, there's no biopsy. But if you really look at it from future standpoint, let's think about it in a way that SEMA was approved. Let's say, we think resmetirom is going to be approved first, but let's say SEMA is the first drug that is approved on the market. Let's say now we are in a world beyond the liver biopsy. What future clinical trials might look like. A drug that is efficacious, and now you're adding to it and using NITs to see improvement in the fat, fibrosis, and inflammation beyond liver biopsy. So I think this design will not be foreign in the future, will not be inadequate, rather it might become the standard of care for NASH clinical trials, including and hopefully registration clinical trials. Of course, I'm not saying those are the right NITs. This needs to be determined. This needs to be a little bit more work. But I think that the study was futuristic in a way or another.
Mazen, I totally agree that the duration was short. And we see that even with the weight loss with semaglutide, about 7 to 8%, as opposed to 16-17% that we saw in the step 1 program. This was, with the higher dose 2.4, which was indicated for obesity. So we anticipate that these patients will continue to lose weight, and maybe that will enhance efficacy. The other point is when we looked at other fibrosis biomarkers, including ELF and MR elastography, we did not see a signal. But again, I think this has to do mainly with the duration of the study. So 6 months, to see change in MRE is probably too ambitious and not realistic. I think, the exciting thing for me is to see rates in the 90% and that 30% relative reduction. And really, I'm hopeful that we get to a point with these combination therapies that we start talking about a cure for NASH in some patients, where there's really no evidence of the disease whatsoever, where there's not enough liver fat to call it NAFLD, where the ALT and the AST are normal and the liver stiffness is back to a normal range. And we can tell, some of these patients that, hey, at least for the time being, it seems like there's not anything to call NAFLD anymore. So that's the hope in the future.
That's great. I'd like to revisit Mazen's comment on the futuristic aspect of this trial. And clearly, the short duration does limit you with the number you repeat these biomarkers. But one of the weaknesses in histology, we have a start and we have an end. We have 2 time points over, let's say, 48-week trial. If we move to the next level and do NITs, Mazen, how often would you actually want to see these NITs being repeated to actually see, a time response that maybe we can learn from too? What do you think is feasible from a financial aspect? Also burden for the patient, how often do they have to come back to the study center, go to the MR or something? What's your view on this? How often are we going to get this in a 48-week trial?
That's a big question, Jorn. I will try to use an example, which is this trial. Let's say I want to design it as phase 2B. Now, I'm not going to say phase 3, so I don't get in trouble with certain people. Again, we need more evidence. We need to correlate with outcomes, and their works are being done with Litmus, Nimble, Neil, NIT, and others on that. But let's say this study, if I want to design it in this patient population, I will probably do it for 9 months or a year on these patients, let's say 2B3, and I will get baseline 6 months for interim analysis and on end of the year. I would think that eventually we're going to reach a combination of NITs, meaning 3 to 4, that will determine both change in NASH and fibrosis. And we have examples now. Again, I'm not trying to sell anything or bias toward anything, but let's say MAST or FAST. We get them at baseline, 6 months interim analysis, and then end of the trial, and show that multiple NITs are heading the right direction. And many of them now are showing correlation with outcomes. And we can show eventually that the improvement in these NITs lead to improvement in outcomes. And I think, realistically, from cost standpoint, I think 3 time points within a year could be sufficient eventually. Of course, with smaller trials with a proof of concept that you really want to see the needle moving on anything or not, you might condense that with shorter duration and more NITs to have more confidence in this trial or the MOA.
I think you also, Naim, when you were talking, actually showed a really good benefit in or reduction in the AST, ALT and FibroScan over that short period. I think the ability to show real-time results to patients short-term really is a motivator to continue lifestyle modification in addition to the medications and to stay on the trials, but also in going futuristic, to be able to engage people in standard of care as this becomes that type of future quite quickly. That ability to show real-world results and how it benefits, even small changes and those medications, has the potential. And I think some of the outcomes that you described fit directly into that short-term reinforcement of it is working. That sounded to me as a huge benefit that you can take into the real-world patient population and get immediate turnaround on their behaviour change. Because we do want to reinforce that any of these treatments in combination with lifestyle modification, because we might be treating one person with a drug, but we're treating the whole family because a lot of family members may be affected by the same condition and genetics. So it's more of a holistic with combination.
I totally agree with you, Louise. And the other point is also to look at futility of a drug. We anticipate to have FDA-approved drugs that will be costly. And I think probably 6 months is a reasonable period of time. I mean, when we look at A1C for diabetes, we do it every 3 months or so. There's no reason to do it more often than that. And the same with, doing MR elastography, PDFF, looking at futility and response to treatment. We'll learn from the phase 3 trial designs before we have the drugs on the market. But for a drug like resmetirom, you would anticipate reduction in MRI, PDFF, ALT, lipids, and you're going to have patients in clinic that you assess at 6 months and don't see any of the above. And in that case, the drug is futile and there's no reason to continue if you're not reducing your liver fat and your ALT is the same as you started. And I think 6 months is fair. Anything less than that would be too short. And then if you see some promising efficacy at 6 months, you said it also earlier that we have to make sure that response is not plateauing and that we continue to see improvement at 1 year. And then maybe after that, we start doing yearly assessments. Of course, patients with cirrhosis will need different monitoring. This is for non-cirrhotic patients.
Can I just ask, on the futility question, if you were to be able to look back in the clinical trials that you're doing now to pick up clear signs of futility at an earlier time point, would that be a benefit given the cost of these medications?
Yeah, I think it's absolutely needed to realize also when to say a medicine is not working, because when we look at the histologic response, for the most part, we're between 20 to maybe 60% response, whether you're looking at NASH resolution or fibrosis improvement. So there would be non-responders, and this is easy with a medicine like semaglutide, right? If you've been on it 6 months and you haven't lost weight, probably it's not going to be effective as a treatment by itself. I think with resmetirom, we're learning the importance of MRI, PDFF, and with every drug, there will be a combination of biomarkers, as Mazen said, that will help us decide when the drug is working and when it's not. But we have to think about it both ways. Obviously, we want the drugs to work for everyone, but also we have to think about the cost to the healthcare system.
So, Naim, first of all, you described this as your baby. At the risk of sounding like I'm saying something facile, your baby's really beautiful. This is a beautiful baby. Having spent my life in a whole bunch of drug areas, none of which were NASH, there are some underlying principles here that make a ton of sense. When you look at the pharmacokinetics of drugs, it turns out that the level that you require to get to peak efficacy is whatever it turns out to be, or significant efficacy. And then above that, the rate of safety and side effect issues rise a lot faster than incremental efficacy does. So there is a compelling logic that goes, if I can use lower doses of 3 different medications, that should have the effect you noted on safety and side effects in addition to whatever value it has for efficacy. And I think that's great, number one. Number two is, I remember the first time I saw a map of the liver in the context of NASH, I was struck by exactly how many different places there are that you can attack. We talk about the idea of taking shots on goal. The problem is the liver has 8, 9, 12, 15 different goals. You're not going to want to shoot 4 pucks at the same place. You're probably going to want to invest in 3, 4, 5 different therapeutic targets. So if you can do that at low dose, then you wind up with a safer world and you wind up with a better shot at meaningful efficacy because you don't need to hit every target with every patient. But, when you talk about a 95% estimate in 3, that says to me that you're getting at least one strike on each target from 90% of your patients, which is pretty exceptional if you think about it. Hey, am I reading that right?
Yeah, I think you nailed it. Cilofexor at the 100 milligram caused more issues with pruritus, increase in LDL. So using a lower dose is helpful in terms of mitigating the adverse events, but we're still seeing potentially some of its anti-fibrotic effects, which is exciting. We know from the obeticholic acid data that there's a robust effect on fibrosis regression with the FXR-agonist. So if we extrapolate this with cilofexor and show that we can mitigate the main adverse events of pruritus and increase in LDL, I think that's exciting. Firsocostat is a very potent medicine, and it's not the only one out there. So there are other what we call acetyl-CoA carboxylase inhibitors, ACC inhibitors, and all of them have a very potent effect on reducing liver fat, and they all share the issue with increase in triglycerides. So in terms of defatting the liver, we are seeing the efficacy, and then adding the semaglutide is helping with mitigating the increase in triglycerides. So as you said, it's both efficacy and also mitigating adverse events with using lower doses of the drugs.
It was very obvious, Roger, if I cut in, it was a very obvious attempt. And I think there's a lot of compounds that could potentially be combined with semaglutide because beneficial effects. And congrats again to both of you to setting this trial up and getting it running. I mean, one of the challenges, and maybe you can give us a little bit of insight, could of course to get two companies talking to each other and being ready to join forces here in this field and, going on this development road together. From an external perspective, that must have been a challenge, or maybe we can learn, and because it's great from the science, but of course that's also a challenge from the development aspect of drugs, right?
Yeah, I think we have to give Gilead and Novo here the credit. They had a partnership on that, and I really wish we can see more of that. A lot of drugs now are, and I totally get it, that they, especially companies that not the size of Pfizer or Novo, they want to get to their approval line and market the drug. So maybe I can call on the larger companies to start partnering with those and tell them it's okay. Let's start combination now. Your drug, phase 3 drug or phase 2B and then 3 will be done way before our combination study is done, and it will not affect your approval. So, I understand there's financial part into that as well from smaller companies' perspective, but maybe older companies or the bigger companies can help the smaller one, even if they don't want to acquire them. That's even actually more preferred in a way or another, that if, it's not time to acquire any other company, let's at least work with them on combination now. Since we're talking about combination. So we all know on this podcast and many of our listeners that this is a field of combo, the combo of NITs, staging the disease and monitoring it, and eventually combination of drug like metformin and SGLT2 in diabetes and metformin and GLP-1 in diabetes.
Roger, probably you can speak to this, but no one has figured out what to do with NASH yet. So I think the companies need each other. In hepatitis C, we have precedent when it was very difficult to treat, and we have drugs from different companies that were combined for a while until each company figured out their own combination, and that collaboration stopped. So looking into this with the same lens, no one has the answer. Every drug has a liability, I would say, and that's why we see the potential for combination. So in my mind, how do I think about it? If your drug has efficacy on NASH, but we don't see clear signal on fibrosis, then you want to partner with another drug that has some signal on fibrosis. If your drug is causing issues related to the metabolic syndrome, let's say you gain weight on the drug, like lanifibranor, which is a great drug in terms of the hepatic efficacy endpoints, NASH resolution, fibrosis regression, but they do gain weight. So what can we do? And this is what Inventiva announced now in a new trial called Legend, where they're going to combine their lanifibranor with an SGLT2 inhibitor called empagliflozin. And the idea is that with EMPA, you're going to lose a little bit of weight, so hopefully that will mitigate the weight gain with lanifibranor. It will have also a defatting effect on the liver, potentially decrease liver enzymes. We don't have a lot of histology data with SGLT2 inhibitors, but I think at least mitigating the weight gain and helping with defatting the liver will be desirable. You can also see a scenario where a drug that makes you gain weight is combined with the GLP-1 agonist, and it could be the pill form, so oral. It doesn't have to be the injectable. Although the weight loss is a little bit less, you can at least mitigate the adverse event of weight gain with another drug. Same thing with dyslipidemia. Again, if you have a drug that increases your LDL, you want to combine it with another drug that has maybe some liver efficacy, but also can lower your LDL to mitigate that adverse event. We saw this with NGM, for example, with their FGF19, where there was an increase in LDL, and almost every patient was started on a statin, and that was mainly to mitigate an adverse event. Again, it's the same theme over and over. You're looking at increasing your efficacy and mitigating the adverse events.
Let me jump on this for a second. I'm just going to be a straight shooter here on a combination that you just mentioned. With a drug with such a good efficacy like lanifibranor and you gain weight, I was hoping to see it combined with a GLP-1 where you lose weight, not with a drug, great drug, but not as effective with weight loss as a GLP-1 or dual GLP glucagons. So that's when we really, as researchers in the field, and we can talk about this, I thought Roger also wanted to talk about it, the right combination. So if you ask me what's the right first combination in F2 patients, for instance, with Lani, it will be a GLP-1. We also have to, in terms of the combination, we have to also mention that a lot of drugs now are metabolic drugs, FXRs, PPARs, you name it. And there's still room for anti-inflammatory, anti-fibrotic drugs. They don't deliver as much when it comes to proof of concepts and 2A studies, especially with PDFF, but that's not what they're meant to be. So we should not bypass them or forget about them. They will work perfectly in combination, the anti-inflammatory and anti-fibrotic. So this is another untapped area that will benefit greatly from the concept of combination.
So, Mazen, I'm glad you went back to Legend, because that was what I wanted to do. What I found so fascinating about that particular combination is, as we hear more talk about focusing on cardiovascular mortality and even on kidney disease, when you talk about SGLT2s, you talk about diabetes drugs that pretty quickly demonstrated value against kidney and cardiovascular nephropathy and against cardiovascular mortality. So the question I had about Legend was, okay, exactly what are we trying to do here? Are we trying to develop the best NASH drug we can, where I think your point obtains, or are we trying to develop a multi-system metabolic drug that affects NASH patients and others as well, in which case Legend, at least it feels to me like it makes a lot more sense. The question that raises is the law of unintended consequences, which is every time you put things together, you get something good and something less good. So A, I want to go back to that, your comment on Legend, but B, I want to ask, at what point do we start to worry or at least consider the things we don't know and the potential for unintended consequences in combinations? Do your comment on Legend first, and then whoever wants to jump in on the other one.
So it's definitely a multi-system disease that you need to, first of all, probably reduce cardiovascular mortality and liver mortality together, and make sure you don't get other organs damaged that, we know the kidneys and other things with NASH. And that's why I exactly commented on lanifibranor. Again, this is me being a straight shooter. I would have loved to see it more with a GLP-1 because it will counteract the weight gain you get it with lanifibranor. It's not that much, but still, it will help with it. That will be my first choice if I had to choose, and it will have multi-systemic effect. As you know, GLP-1s, they have cardiovascular protective effect, they have weight loss, and thus they will pull the other metabolic features to the right direction. Again, I'm not trying to bash the SGLT2s or it's underestimate them, but in a drug that is making you gain weight, add another systemic drug that is going to make you probably lose more weight.
But Mazen, with the SGLT2 inhibitors, you lose about 3 to 4% of their total body weight, and this is what we saw with lanifibranor in terms of weight gain. So I agree with you that it'll be nice to even see some weight loss, but I think the idea here is to just mitigate that weight gain and just become neutral. Because what we know with the weight gain with lanifibranor, that it's actually good weight gain. Basically, you are redistributing the fat from visceral adipose tissue to subcutaneous adipose tissue. So it's not a bad way to gain weight, but obviously our patients do not want to see their weight go up. We've known this with pioglitazone. Actually, if you gain weight, it means the drug is working for you. So I think the idea here was just to mitigate this. Otherwise, SGLT2 inhibitors will hit on every other point, right? Because we'll see some defatting of the liver, reduction in liver enzymes, improvement in cardiovascular outcomes and kidney outcomes. There's a very good rationale for this one, and I think there's also a good rationale to do a GLP-1 agonist. So both of them make a lot of sense.
That makes sense. Now do me a favor, let's move on to the other thing that I'm curious about, right? If 3 is good, are 4 better? Are 5 better? And at what point do we start to think about the unintended consequences of combinations where we get things out of them we don't want and didn't expect, whether good or bad, and how does that affect thinking now, or how is it likely to affect thinking in the future as we've got more modes of action that make sense, that are proven?
Let me take a jab at this. So what we know now that probably one will not be enough, or I shouldn't say probably, most likely one will not be enough. And there are multiple scenarios. So to me, the least amount of the drugs, the better it is. If they are all oral, which is going to be probably unlikely, that's even preferred. But also, I think there are pockets of patients that there will be an art in treating NASH patients. For example, the cirrhotics, even the F3s will be different from F2s, and F2s will be different from F3s at one point, and F3s will be different from F4 when we use the combination. So if you ask me what I would like to see in the future, I would say the perfect two in a personalized medicine approach with the least side effects.
To extend that, we might not find the perfect patient for that in the phase 3 trial because those might not be big enough. We got to get the drug approved. We got to then look at phase 4 and actually maybe start seeing some real world evidence on certain subcategories that, well, clearly it's not as controlled anymore as in clinical trials, but I think it's important taking this forward and then defining it. So you might, even if we get the drug approved, might not have the answer, what's the best patient for that drug yet, and I think it'll be important to stay on top of it and be open-minded in terms of how we move these field forward. Now, of course, the big challenge is going to be to get the first drug approved, but after that, I think we'll have many more answers in terms of what's the best fit for which patient.
Totally agree. And this will be a practical way to look at the perfect combination.
I think we have an unprecedented situation where we really have no drugs approved and we're thinking about combination already. In diabetes, things come in waves. So, we've had metformin, sulfonylurea, then you had the GLP-1 agonist, then the SGLT2 inhibitors, and then you find a place for them. But this is very unique. If you think about what's coming, hopefully in the near future, we have two drugs that are maybe getting FDA approval. One is obeticholic acid and the other one is resmetirom. And if you think about them, obeticholic acid has efficacy on fibrosis, but it has the issue with the increase in LDL. And then resmetirom has efficacy on NASH, but we haven't seen fibrosis efficacy in terms of histology, although we've seen some NIT data that looks pretty good. But let's say resmetirom and Maestro-NASH just shows NASH resolution, no clear benefit on fibrosis. Resmetirom has great effects on dyslipidemia, including lowering LDL. And both of these drugs, if you look at fibrosis regression, we're talking about 20-23% range. If you look at NASH resolution, maybe 35-40% range. So it makes perfect sense from day one if we have two drugs to look at that combination of a THR beta and an FXR agonist, especially in a patient who's starting with F3 NASH. This potentially could be one of the first combinations that we will utilize, and obviously we will have data to support this. But some providers may have two drugs to choose from, and it makes perfect rational sense to combine them. And I think this is the rationale for the new study with the Terns Pharmaceuticals. So they have their own THR beta and they have their own FXR agonist, and they're doing a combination therapy trial now with these two agents. And I'm very excited to see what happens in terms of increasing efficacy, but also mitigating the usual side effects from an FXR agonist.
All makes good sense. And let me go back to the question about 2 versus 3 that Mazen raised we've been on. If I understand correctly, we've got three possibly separate issues here. We have an issue of fibrosis, we have an issue of liver defatting, and then we have a metabolic issue. And the metabolic issue and defatting the liver can overrun each other, but they're hardly identical, if I get that right. Which might say that 3 is the right number, not 2. It also so happens that Naim, the trial that we're talking about, cilofexor, is a 3, each of which is targeted at one of those places. But in the end, is there a compelling logic for 2 versus 3? Is it just going to depend on what the drugs are? And then I'll go back to my question, which is, if you add kidney as a 4th, or cardiovascular, is there any time that 4 is the right combination? But at some point, the law of unintended consequences is going to eat you alive. I'm just kind of wondering if we have any sense of where that is.
No, so there's inflammation, all this. Again, I'll go back to the least amount of the medication. We know we're not going to get away with one only, and I'm sorry to hijack the answer again, but when you're going to try your best to do 2 and not 3. And the two will be hopefully having effect on the liver and other organs, heart, kidneys, obesity, and the liver one hopefully will have effect on fibrosis or even better inflammation on fibrosis because it's the most prognostic one. So adding is, we should go up from 2 to 3 when we really cannot get away with 2. And 4, we should make it really harder because it's a cost, multi-system. We know what's the disease prognosis. We know what is the most important factors in the disease. So we have to be realistic and not trade with the cost and side effects of medications. And polypharmacy, don't forget these patients are already patients that they have other issues, including diabetes, obesity, dyslipidemia. So hopefully one of these drugs will hit 2 birds with 1 stone, or whatever you say, to make sure it's being used for both liver and other indications that the patient has.
But let me be the devil's advocate here and just say that there will be patients that will actually just need one drug. I mean, if you have someone with F2 NASH and liver stiffness on the lower end for F2, and let's say you start them on semaglutide, and now they're losing weight, they lost 16-17% of their total body weight. You hit already the cardiovascular outcomes, the weight loss, the dyslipidemia, and insulin sensitivity improvement, and you see that your liver stiffness is coming down on NITs. Why would I need a second drug for this? The only piece that's missing is maybe the fibrosis. And the fact that we didn't see it in a phase 2b doesn't tell me that, semaglutide will not have anti-fibrotic effects. In fact, we see this with bariatric surgery clearly, that fibrosis regression is the rule as opposed to the exception. So I see some patients that maybe they just need one NASH drug plus lifestyle intervention and they'll be okay. Maybe not for life, but for a long period of time. And if we see signs of progression, then maybe we can add a second drug. But I don't think we need to think, we're going to do 2 drugs for everyone and then start talking about 3 and 4. And keep in mind, many of these patients will come to us on a statin and a fenofibrate and potentially a GLP-1 agonist as part of treating their metabolic syndrome.
Naim, I would totally agree with that. I'm not saying 2 is going to be the case scenario. Rather, I'm trying to minimize the number of drugs we use. And I'm in total agreement with what you said. And it goes back to previous comments when I said it's going to really vary per patients. And we just have to use our clinical sense in managing these patients. And the example that you gave for F2 is, I'm in total agreement with it.
Can I ask a dumb question in relation to clinical trials? And
Louise, I just asked 6 dumb questions in a row. You're going to have to go a long ways to catch up. Go ahead.
So bringing in your point, Naim, that these patients are going to come to us on other medications. If you were to write a new baby, because you only had 108 patients in this baby, and therefore the diversity isn't necessarily going to meet the criteria that we currently have, and we need to know which these drugs work in different populations. But if you then said as part of to alleviate the problem of the 16-week lead-in and finding the right patients who respond to SEMA. And obviously this is a licensed drug around the world in multiple different areas, weight loss, obesity and diabetes. If you said your inclusion criteria for a trial with the other 2 medications was to have been stable on semaglutide for more than 6 months, would you be looking at the ability of semaglutide being implemented around the world in mass populations to be able to get that diversity that you would need in different ethnicities, for example? Is that a way to approach these clinical studies, to actually put that into the inclusion criteria? Because we know they come with on medications. We talk about these 3 medications, but they're on a whole host. So we use that as the benefit to developing the trials because they are a cohort of patients.
Just so I understand you 100%, you're saying that we have patients that are already on semaglutide and they're on a stable dose for 6 months and then they're coming to us and we add 2 drugs from Gilead, cilofexor and firsocostat. Is that the design you have in mind?
Yeah. Would you be able to write that? Because, of course, semaglutide is used around the world in a lot of the populations that we target. Because they don't necessarily fibroscan or use FIB4 or any of the other techniques, we don't know the level and prevalence of F2 to F4 in those populations because it's not looked at.
Yeah, it'll be interesting. The concept is not foreign. So like in hepatitis B studies now, we we require that the patients are stable on their hepatitis B medication, which is usually a tenofovir version for a while, and then, we add new medicines and we provide them with the tenofovir as part of the trial. So it could be done. My main concern is, are these patients are already non-responders to semaglutide because they're showing up with NASH and F2, F3 despite being on semaglutide for an extended period of time? And are we going to see an added benefit in the trial with semaglutide, or are we just testing the two other drugs as, a treatment for NASH, and we already selected a population that doesn't respond well to semaglutide? But I think, that's reasonable. But speaking of babies, Louise, I'll tell you my next baby that we're working on already with this triple combination. So here at the liver meeting in London, the EASL ILC, and you saw the data with Novo in cirrhotic patients, right, with the semaglutide monotherapy. And there was no clear benefit. I mean, maybe on some of the AI data we saw a signal, but at the end of the day, I would say it was a negative trial, and semaglutide monotherapy did not help with NASH cirrhosis. So the next trial that we are doing with Gilead and Novo Nordisk is actually looking at over 400 patients with NASH cirrhosis, and then using this triple combination, and we're treating patients for 48 weeks, and the primary endpoint is fibrosis regression on biopsy. So this is the next baby. It's going to take probably another 18 months to see it being delivered. This is like an elephant baby. It's a big one. So the pregnancy is about 3 years, but we'll see. I think this will be very important for the field because, obviously NASH cirrhosis is the highest bar, and if we can actually show a regression of fibrosis, that would be very impactful. But I'm optimistic because number one is semaglutide is a powerful drug. There's some data emerging now, and I'll be able to share more with you guys on the effects of bariatric surgery on NASH cirrhosis, because this has been a very controversial topic. Do you get improvement in fibrosis? So give me about 6 months or so, and we'll discuss this, but we have a new study coming on that. I'm encouraged by the Atlas data, looking at firsocostat and cilofexor, 56% cirrhotics, and there was some signal on fibrosis regression on biopsy. So we'll see, but this is again probably 2024 before we have the results.
Can I ask about one other baby that hasn't been mentioned yet and may not even be a baby? It may simply be a glimmer in my eye. Mazen made the comment a while ago that it would be great if all these agents were oral. There is an oral sema, right? It's not as efficacious as the non-oral sema, for want of a better term. But could we look, for example, in a situation like that, at making that a part of a combination package, an oral combination package? And are you aware that whether that is or isn't being done right now, that you can say?
I think the main concern from Novo was that there are no GLP-1 receptors in the liver, and that the mechanism of action in terms of NASH resolution is mediated by weight loss. So they wanted to go with the highest weight loss that you can achieve. But I totally agree, Roger. That makes a lot of sense. And, Novo is not the only company with an oral GLP-1 agonist. So there are discussions, and there are other companies that have oral GLP-1 agonists that are being looked at as monotherapy and potentially as part of combination therapy. It makes a lot of sense, and the weight loss is actually better than the SGLT2 inhibitors, although it's not as good as the injectable GLP-1 agonist. And there are other companies, obviously, with dual agonists, as Mazen mentioned earlier. There's Altimmune, there's Lilly with tirzepatide, and now we are seeing weight loss in the 20-22% range with tirzepatide. And then Novo just had a press release also with the new combination called Cagri-Sema, and in that one there was more weight loss than semaglutide monotherapy. It was a relatively short term, 32 weeks, but definitely more potent than sema monotherapy. So there's more to the story. We didn't discuss also every combination trial. Novo has another combination trial that they're doing with Cagri-Sema plus an FGF21 agonist, which would be similar to the Akero and 89 Bio. So there's a lot that's happening in that space. Part of it is let's see how much weight loss we can induce, but also let's look at liver targeted effects in terms of defatting liver, NASH resolution, and eventually we want to see fibrosis regression with these combinations.
We're heading to the bottom of the hour, and I guess to steal Mazen's metaphor, the first thing I want to do before we go to final question is simply ask the Matt Damon of hepatology if he has any insights on either how Naim can manage not to gain weight during a 36-month pregnancy, which sounds challenging, challenging to me. Or generally, any insights on how to get any of these things sorted out faster or more efficiently than we're talking about today?
That goes to me, Roger.
And if you don't like that question, answer whichever one you choose, okay?
No, it's fine. Just listening to the last comment of Naim, I was reminded we're not developing a weight loss drug for patients with liver disease. We're developing an anti-metabolic, anti-fibrotic drug in patients with severe steatohepatitis and fibrosis. So it's not all about weight loss. And I would want to see, it's good to crank up the amount of weight that can be lost, but I think we're all looking for that modifier that maximizes the fibrosis regression or turns off steatohepatitis in these patients. So that's point one. The time it takes to resolve some of those changes, some studies have shown very quick results, even on biopsy. Remember 12-week biopsy studies showing some improvement in fibrosis. So I think still, after all these studies, we're seeing some heterogeneity across the clinical trials. And as I mentioned before, I think part of that heterogeneity we're just going to sort out in the bigger cohorts when we treat more patients and have drug available in clinics. But beyond that, I think, the approach is reasonable. And I'm just like to congratulate both Naim and Mazen again for bringing this one over the finish time, although it took some while to get published, but good things are close on the horizon.
Amen to that. Final question to everybody. I'm going to ask everyone to take a stand. 7 years from today, what percentage of patients being treated for NASH fibrosis, non-cirrhotic and then cirrhotic, will be treated with combination therapies? 7 years, because I think it gives enough time for the market to sort out a little bit on monotherapies as well. Brave one, go first. We're going to put it in a time capsule. 7 years from today, someone's going to open it and say, gosh, were you guys smart?
I can give you a quick answer here. I would say the majority of patients with NASH cirrhosis will be on combination therapy. I think the majority of patients with F2 fibrosis, maybe F1 by then, will be on monotherapy. And then F3 is going to be probably 50-50 with the trend toward, adding more agents over time to prevent progression to cirrhosis.
I have sometimes to disagree and argue. It's not because Naim is wrong. I think in 7 years, we will not get to combo for NASH patients just from regulation, partnership, logistics, and all that. I will be happy in 7 years if I'm using something on NASH cirrhotic patients.
I was going to jump between the two and be slightly positive in the fact that I agree with Naim. If we get there, it would be great. But I do think that maybe 80 to 90% of cirrhotic patients will be on a combination therapy, purely because they're the ones in greatest need, I think, because of the cost and the advantage in F2 fibrosis, then probably maybe only 10 or 20%. But I think the F3 and those identified as fast progressors will go on to combo therapy because those, again, are the ones who are in need and progressing more quickly. There will be stratifications of who gets access to medications. We haven't even got to the stratification who's even going to get located and found yet. So we got a number of hurdles to jump. I'd like to think we're there in 7 years. I'm probably more with Mazen on that there will be a lot of hiccups in the way, but hopefully there'll be some fast tracking and some great outcomes. And this baby being delivered by us hepatology gynecologists and midwives is actually not a bad one.
I have a question, the final question. What is Matt Damon's most famous movie?
Well, wait a second. Matt hasn't answered this. Jorn has not answered this question, and then we can go to your point, which I'm sure is going to be
sure. I'll invite you guys over to my next movie set, and we'll hang out. You can be the groupies, and I'll be doing the work. Anyways, I like The Bourne Identity. How about you, Mazen?
I think I've seen, believe it or not, me being a fob, and a lot of people would know what's a fob. I have seen these series recently, and I quite enjoyed them. And I honestly thought of you when I was looking at him.
All right, to bring this back to the actual content of this podcast, Roger, this has really been hijacked by a movie fan here. I think there's multiple hurdles, and looking at the way where we identify the patients, we got to still do a lot of education on partnering physicians and really educate them about the NIT and identifying these. Even if we have a drug approved, of course, it's going to be helpful to expedite that process. But we got to get the at-risk patients identified in endocrinologists' practice and primary care practices, using the right NIT, getting the NIT reimbursed, and then having access to the drug. And adding 2 drugs in these is going to be a perfect way to round this off. But I think I'm still a little concerned that we will miss a lot of these patients. They don't have access to diagnostics even.
So a couple thoughts. Right, number one, you and I agree with you that diagnostic within 7 years is going to be a challenge to get appropriately scaled, particularly if we've got to pull it back into primary care, endocrinology, et cetera. Number two, I'm mindful of diabetes and actually hypertension also, where we went from different competing monotherapies to different forms of step therapy, if you will. So step therapy actually is a path to combination therapy that doesn't require single agents that have dual modes of action in them. So my guess is that in 7 years, we're less likely to see a bunch of 2 or 3 drugs in a single package, more likely to see first line, second line, third line add-ons where you wind up fundamentally with a combination of therapies, even though it isn't where you started. And if you think about it that way, my guess is there's going to be a lot of that in cirrhosis, a bunch of that in F3. I'm not sure my numbers are exactly where Naim's are, but they're not off by much. That's going to be about timing, and less so obviously in F1 or F2, where people are going to believe a single therapy can get enough of it done. That would be my guess. And here we really are guessing, but if we're doing this podcast in 7 years, or if a successor is, I will make sure that they pull this out and see exactly how much we missed by. The winner will get to decide who besides urine should be patterned after a movie star, and which star that would be. Mazen, I would be very scared if I were you. And with that, let me thank all you folks. This has been a great conversation and frankly a fantastic episode to come back to from vacation. So Naim, we don't get you here often enough because you're too busy, but I want to talk to you about what we can do to change that because your contributions are just so spectacular. Mazen as well. The rest of us, we're here all the time.
No, it's always a pleasure to be on this podcast, and I'm happy to contribute as much as I can. Just the specific time is always killing me because it's in the middle of the clinic, but I understand with Jorn also being in Germany, it's not easy to find a good time, but we'll figure it out. If you give me enough notice, I can always block my schedule. I get asked about this all the time, so it's important to stay on the podcast. And the other thing I wanted to mention is I'm organizing a basketball game at AASLD. So Matt, Jorn, you play basketball or no?
I'll give my best shot. I won't do the hoop jump, but I'll hit a couple of threes. So let's try.
Like where? And there's nothing around that thing in DC.
I'm working with Rachel Zayas, so she's going to organize it. She's a basketball player. So I don't know if you want to leave this on the podcast, but maybe we can make an announcement that there will be a basketball game at AASLD in Washington, DC in November 2022.
Well, so Naim, my expectation is that Rachel's going to be part of the coverage team for Paris NASH, so we might actually wind up coming back to all this with her to see what she's got to say about it, okay? And thanks, you guys. This has been great, all of you, and we'll be back next week talking about Paris NASH and about the NAFLD Summit, and I'll be back with Business Report in a few minutes.
And now for the Season 3, Episode 43 Business Report.
Slava Ukraini, Heroyam Slava, glory to Ukraine, glory to heroes. Last week, which was the 6-month anniversary of the start of the war and the Ukrainian Independence Day, I was on vacation and had pre-recorded this opening comment. This week as I return, much is happening on both sides of the war, with Russians conscripting troops up to age 60 and Ukraine reportedly preparing a counteroffensive. I continue to commend the amazing courage of Ukrainian forces while hoping all this will come to a just end soon.
First, a note of thanks.
I want to start by thanking the entire team, but particularly Louise, Mike, and Jake for producing last week's episode and online support so that I could unplug from NASH Tsunami for a week. You folks were superb. You have my deepest gratitude, and now I'm back and revved up for an exciting autumn.
Our specialty podcasts are out in distribution and doing well.
As of this afternoon, we've tracked well over 100 listeners for the 2 pilot series, the Rising Tide in Diabetes and the CME series on use of NITs. You can access the entry questionnaire for either program by looking on the top ribbon of our website. Also, we'll begin advertising for the 2 series later this week. We welcome all feedback about the programs, episodes, advertising, choice of topics, whatever you wish to comment on. We await your feedback.
Coming this month, NASH Tsunami covers Paris NASH and the NAFLD Summit.
Next week's episode will preview the month's 2 major conferences, Paris NASH on September 8 and 9, and the NAFLD Summit the following weekend, I believe the 15th through the 17th. Each of the 2 weeks after that will be devoted to one of these episodes, and then after that, returning to the question of drug trials failure and what we've learned.
NASH Tsunami downloads remain strong through a slow news time.
The last full week while I was on vacation saw over 1,700 downloads, which is the total we have through 5 p.m. Eastern U.S. time today. That's slightly less than we had 5 to 6 weeks before that, but far better than we did before that period. And I anticipate that number for this week might kick up in the next day and a half and again in the next few weeks as we cover the conferences. So with that, I'm off. Thanks to the crew, Magic Mike, Eric, Steve, Jake, for everything you do, and triply for what you did last week. Remember to look for the ads on the CME episode and the diabetes episode by the end of this week. And as always, thanks to all of you for making this such a worthwhile labor to pursue. Stay safe, surf on. If you're in the States, enjoy your Labor Day weekend, and we look forward to seeing you again next week on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.